Hi, good morning. Thanks, everyone. Tanya said, I'm a postdoc at Carnegie Science in the Baya and Grossman labs, where we're studying the phototrophic mat community of hot springs in Yellowstone National Park. So if you heard me at the JGI user meeting, I'm actually going to give a slightly different variation for here and focus on some of our data on the hot spring Armatinomona, which I might refer to as ARMA because it's a huge mouthful. But first, let me introduce you to our system. So the hot spring microbial phototrophic mats form in the outflow channels from hot springs. So as the water cools, as it flows away, between about 70 degrees Celsius and lower, you form these layered phototrophic mats. If you take a core sample, as you see in the middle, you can see the nice layers at the very top. We tend to be dominated by cyanobacteria, and then underneath layers of various anoxygenic phototrophs and other heterotrophs. And we'll go into a little more detail about them in a minute. And a lot of times people think about thermophiles. We really want to focus on how do they survive heat, but today I want to talk to you about some different stressors that are found in this environment, and that is the stresses involved in the day-night cycle. So during the day, these mats, as they experience light, the oxygenic phototrophs produce tons and tons of oxygen. It can get up to 800% air saturation in certain parts of the mat. pH also increases during the day as carbon is fixed, and there's very little H2 around. At night, the mat quickly becomes anaerobic. pH also decreases as fermentation happens, and we do also observe some hydrogen at night. So these are really two very different regimes depending on the time of day. So our question was to better understand how does the mat respond to the day-night cycle in a more community context. So our approach with support from a JGI CSP was to do hot spring metagenomes from 34 different mat cores from a variety of different temperatures from 2 springs. From these metagenomes, we assembled in bin to mags. We got around 800 mags. For 60-degree samples in particular, we did RNA-seq metatranscriptomes for every 2 hours from a 24-hour time point, and then mapped those reads back to our mags, and that's the data I'll show you today. So just a very quick overview of our mags that we got from all of our metagenome samples. We recovered over 800 high-quality, medium-quality mags from about 71 genera. So there's a lot of data here, but basically there's a number of maybe 10 or so that are really abundant across all of our samples. And then depending on some of the properties of the samples, temperature, depth, we find different other members. But when we go to our metatranscriptome at 60 degrees, we actually only find 8 of these genera are really most active, so above 1% or more of our mapped reads. And I'll be trying to use these same colors throughout the talk to indicate kind of their roles. So first, our cyanobacteria and oxygenic phototroph, we're calling it Synechococcus. There's a lot of taxonomy debate right now. So this is what we think is the main primary producer. It fixes CO2. It also produces oxygen. We also have 5 genera of anoxygenic phototrophs from 3 different phyla. These use a variety of different carbon sources and a variety of different reductants for their metabolism. And there's been a lot of work on these in this mat, and I'm not going to go into them today. But today I wanted to focus a little bit more on our 2 heterotrophs that we were predicting to be aerobic heterotrophs from Armatinomona, used to be called OP10. And the names don't mean too much here, but we're going to call one OTU3-like and one OTU12-like for today. So our 2 strains, so here's a ribosomal protein S3 tree of available genomes on NCBI. And you can see our 2 strains are in red, and I've indicated in the arrows where 2 of the cultured members are. So our strains are pretty distant from the cultured ones, but we do know a little bit about these organisms. The ones that have been discovered tend to either use sugars or tend to be more proteolytic. So from our metagenome, we're able to predict a bit about the metabolisms of our 2 strains. and they are quite a bit different, and I'll start highlighting some of the key features. So first, looking at their electron transport chains, we find both of them are predicted aerobes, but possibly some differences in the exact chain. I'd love to chat with someone about how to better work this out. One thing that was a bit surprising to us is both of these strains contained rhodopsin, so they might be able to use light energy as a supplement. If we look at the TCA cycle and central metabolism, the OTU3-like is missing two key steps, including malate dehydrogenase, although it does seem to be able to use the glyoxylate shunt. This is consistent with what's been described for the fibromonas strain of Armatida. The OTU12-like strain is more of a traditional TCA cycle. And then we used some of the tools Gapmind to help predict some of the carbon sources it might use. So OTU3-like seems to prefer amino acids and also maybe TCA intermediates, small fatty acids. In contrast, our OTU12-like strain, we think is going to be using polysaccharides and small sugars. In addition, it also does have hydrogenases present. So that was a little bit about the potential of these two organisms, but going back to our metatranscriptome, we wanted to know a bit more about the actual RNA-based activity of the environment. So if we take a very bird's eye view of looking at when genes are most highly expressed, so more yellow is the most expressed time point. So of our 8 organisms, you can see there's quite different patterns in peak expression. And the Armatida in particular have really 2 different patterns. Our OTU3-like organism seems to have a lot of genes peaking in the day, and our OTU12-like organism has a lot more of its genes peaking in the night. We hypothesize this might suggest there's differential timing of the preferred carbon sources. Some prior work by Kim et al. Has measured whole mat metabolites and found sugars seem to be a bit more abundant at night, but we really want to follow this up and find out more about what's actually going on in the mat. And then I want to just finish with a couple very quick vignettes about some of the other organisms and genes we observed in the metatranscriptome, because that was a bit more about when are things happening, but we actually want to know what are the pathways happening when. So if we look at photosynthetic reaction centers, we have all 8 of our organisms and the transcripts plotted here. If we look at the photosynthetic reaction centers, they are as we expected and has been observed before, that the Synechococcus expresses its reaction centers high in the day, and all of our anoxygenic phototrophs really express those high at night. We then looked at the expression of rhodopsins, and so our 2 ARMA species do both express rhodopsin, so that does suggest they are possibly actively using the rhodopsin in the mat. We also observed the expression of rhodopsin in 2 of our Chlorophlexota anoxygenic phototrophs. So these guys are going to be able to do both chlorophototrophy and rhodopsin-based phototrophy. And then a final quick story that we found that was a little bit surprising to us was we observed chaperones, I'm plotting here just GROW-EL, seem to have a temporal expression in the mat, in that you can see it very clearly in the Chlorocidobacterium and Roselinea species that these chaperones are really going to very high expression at night. One hypothesis we have is that perhaps this is related to the production of the reaction centers and the photosystems in our phototrophs, but we also recognize there could be other things they're involved in and are actively trying to figure out, are these related to cell cycle? Are these related to some sort of stress rates of protein synthesis? So in summary, we found that 8 genera are really the most active at 60 degrees C in the Mushroom Spring in Yellowstone National Park. We're trying to better describe these Armatida monodota that have been found in the hot springs. And in the future, we want to use more in situ measurements to better understand what's going on and also try to cultivate some of these strains in the lab. So I'd like to thank the organizers and thank my lab and our collaborators and our funding, and I'd be happy to take questions.
Great talk, Amanda. Thank you. So again, for the sake of time, I think we're going to take a break now for the online participants.